I'm gonna introduce Andrew Hsieh, a good friend of mine, and one of those rare people in life that can actually translate translation into human terms that we can all understand. And so today he's going to be talking about his work on the androgen receptor, which regulates a druggable translational regulon in prostate cancer.
Well, thank you very much, Brett. I want to thank you, because you guys have proof that persisters can be good. All right, so these are my disclosures. So my laboratory is focused on understanding the core mechanisms by which the process of mRNA translation can actually drive prostate cancer initiation progression. And we seek to do this at a deep mechanistic level. And the way we look at translation, it's really a complex dance between the protein components that enable translation and the RNA components. And a perfect example of this is when the eIF4F translation initiation complex, which Daniela talked about so nicely, interfaces with the 5' untranslated region. This collision enables the recruitment of the 4DS ribosome, which of course is essential for translation. So with this in mind, today's talk is going to focus on the two aspects of translation, the protein side of translation as well as the mRNA side. And this is really all girded with a current understanding of the evolving landscape of advanced prostate cancer, which over the past, decade and a half, we now know with ARSIs that there's an increased incidence of AR low or deficient prostate cancer. It's a scourge to mankind. So we sought to actually study this in autochthonous mouse models of prostate cancer. And we started with a simple experiment. We took the P10 prostate cancer mouse model, we castrated it to decrease testosterone. We asked, what happens to the tumors? And what we found was that over time, the tumor glands would get actually bigger. We saw increased proliferation, as shown here. But what was really surprising to us is when we looked at the androgen receptor levels, using 3 separate antibodies, it went down dramatically. And so, given the fact that it's already been shown that androgen receptor levels inversely correlate with proliferation, nice work done by folks here in this audience, but also that proliferation positively correlates with translation. We want to understand how androgen receptor activity correlates with translation. So to begin to understand this at a whole tissue level, we utilized a labeling experiment called a puromycin incorporation assay. And what we do here is you inject the mice with puromycin, it gets incorporated in the nascent chain, and then you use an antibody to detect this, and it's a readout for de novo protein synthesis. And what we found is this actually leads to a significant increase in new protein synthesis. It was actually very satisfying. About a few years after we made this discovery, Adam Sharp from the IRC reached out to me with this particular experiment where he used AR to knock down, he used an siRNA against AR, knocked it down in LINCAP 95 cells and showed that that also caused an increase in protein synthesis. Now, given this connection with androgen receptor and protein synthesis, the obvious question is, how does this work? Is there a mechanistic link between the two? Well, we were inspired by work by Nahum Sonsberg's laboratory, which showed that a particular component of the translation apparatus, which we've already learned about, the eIF4F complex, is actually really important for the proliferative capacity of cells. And this particular complex actually binds to the 5' cap of mRNAs composed of eIF4E, which is the cap binding molecule, 4A, the helicase, and 4G is the scaffolding molecule. What's really important, this entire complex is actually inhibited by an acidic molecule called 4EBP1. And when that happens, you actually decrease translation. So we did a very simple experiment. We did immunofluorescence of all these proteins, and what we found was that indeed, with 4A, 4G, and 4E, we saw no difference at the protein level. How with 4EBP1, we saw a significant decrease. Now, this could be because of the model. This is a mouse model, right? No model's perfect. So we looked at the APIPC model generated by the Nelson Laboratory, which does not have AR, and indeed 4EBP1 levels were down. We also looked at all of the PDX models, the LUCAP models, and found a positive correlation between the androgen receptor protein levels and 4EBP1 protein levels, suggesting that AR might regulate translation through 4EBP1. But the question is how? So we considered many possibilities. We looked at translation of 4EBP1, it wasn't that. We looked at degradation of the protein, it wasn't that. However, when we looked at transcription, we found that in all three of those models that I just showed you, in each case, 4EBP1 mRNA levels are decreased when you either castrate or you have low antigen levels. This got us thinking that there might be a causal relationship between AR signaling as well as 4EBP1 transcription. So in order to test this experimentally, we took the castrated P10 mouse, we made organoids out of them, and then what we did was we added back DHT. And what that did was restore 4EBP1 transcript levels as shown here. Now given this link, we thought maybe AR directly regulates the 4EBP1 syndrome. So to answer this question, we teamed up with Yu Chen, who had already done AR chip-seq on the P10 model. And what we found was within the first intron of 4EBP1, there's actually a binding site for AR. And in fact, if you look closely at it, there's an antigen response element. Went ahead and cloned this out into a reporter construct and found that it was indeed antigen responsive. This basically demonstrates that AR can actually control translation through a very important gatekeeper protein, and that's 4EBP1. Now, given these particular findings, we were very interested in understanding, okay, if 4EBP1 levels are low, do we actually get more translation initiation complex? Well, in order to answer that question, we used complementary proximity ligation assays, where we can measure the amount of on-complex formation versus off-complex formation. And what we found in the castrate system was that upon castration, that leads to a significant increase in on-complex formation and a significant decrease in off-complex formation, demonstrating that we weren't just decreasing 4EBP1, but we're actually increasing translation initiation. So if indeed we were actually in increased translation initiation, then the obvious question is, does that control proliferation in our model? Well, to answer that question, we generated a mouse model in which we can add doxycycline and actually induce the expression of a transgene called the 4EBP1 mutant. It's a dominant negative form of 4EBP1 that can inhibit the interaction between eIF4E and 4G, thereby obliterating translation initiation. That's what you see here in the PLA. And when we went ahead and crossed that with the P10 model, castrated them, added doxycycline, we saw a significant decrease in tumor volume as well as a decrease in proliferation, as shown here. Now, given these findings that AR might actually regulate regulons of translation, we wanted to know what they were. And so in order to do that in an unbiased manner, we conducted tissue-based ribosome profiling. So ribosome profiling is a whole transcriptome method to take a snapshot of all translating mRNAs. In doing so, we were actually really surprised to find that it was actually a specific subset of mRNAs that were being translated more. And when we did GSCA analysis on these, we found that one of the subcategories that were enriched were cell proliferation mRNAs. So we did a lot of molecular experiments after that, but what we found was that these particular mRNAs, the reason why they were sensitive to increased levels of eIF4F was because within the 5' UTR, they actually had a distinct cis-regulatory element, which we called the guanine-rich translational element. Now, given these particular findings, it suggests that AR-low prostate cancer might actually be uniquely sensitive to inhibition of translation initiation. We want to test this preclinically. So we used a tool compound called 4E1R-CAT. What this does is it breaks apart the 4E4G complex. And what we did was we treated both AR proficient and AR deficient xenograft models. And this is an example of one right here, where you can see the AR deficient mouse xenograft models was exquisitely sensitive to the inhibitory effects of 4E1R-CAT. However, when we looked at the AR proficient model, it was completely insensitive. And we tested this in PDX models as well. Now, we were really excited about these findings, but we were also humbled by the fact that 4E1R-CAT is a molecule that does not have good drug-like qualities. So in answer to this problem, our lab actually started a collaboration with Cyclica, Inc. to use their AI-assisted approaches to screen millions of compounds that could potentially inhibit the 4E4G interactions. We've already done the screen. We found 131 compounds, which we're currently testing in vitro. So for the second part of my talk, I'm going to focus on the mRNA side of translation, with an eye on the 5' untranslated region. So the 5' UTR is critical for titrating levels of protein within a cell. And it really does so through two different levels of gene regulation. The first is at the DNA level, and the other is at the RNA level. And it does so because at the DNA level, there could be promoters that are encoded within the 5' UTR that enable transcription. And at the mRNA level, there can be cis-regulatory elements or structures that can enable translation or transcript stability. Now, we wanted to ask the very simple question, is how important are 5' UTRs in prostate cancer etiology and progression? And we decided to answer this question going into prostate cancer genomics. And in particular, what we did was we conducted UTR sequencing across hundreds of patient specimens. And what we found, not surprisingly, were there were thousands of point mutations within the 5' UTR. In fact, hundreds of genes had actually multiple mutations in the 5' UTR. But the issue is that these are just mutations. Which ones are actually functional? There were no good technologies to actually study this in an unbiased manner. So as a result of that, we created our own. It's called Plumage. So Plumage is a massively parallel reporter technology that enables us to study any somatic mutation found in individuals within the 5' UTR's native length context. And we do that studying both the effects on transcript levels as well as translation. So when we did this for our genes that had multiple mutations in them, we actually found that about a third of somatic mutations within the 5' UTRs can actually have an impact on transcript levels or translation. So what I'd like to do now is give you a flavor of two of these that we found, because we did orthogonal studies to validate their functionality. One was a transcriptional hit on FGF7 5' UTR. Another one was a translational hit on the CKS2 5' UTR. So first for the transcriptional hit. So FGF7 is an oncogenic protein when it's transcribed and translated. We found this mutation in the transcriptional hit, meaning this hit allowed increased reporter signal. And so we asked a very simple question. We go back to the patient with this FGF7 mutation. Do they have more FGF7 mRNA? And what we found was indeed they did through RNA-seq. But as we looked more closely at this particular mutation, we found it took a regular plain piece of DNA, and that C to T mutation actually created an E-box element. Now, so for those of you who are MYC aficionados, you already know the answer to this. But for those of you who are not, the E-box element is what MYC, the oncogene, binds to. As we know, MYC is highly deregulated in prostate cancer. So we wanted to ask the question, could this mutation enable the binding of MYC to an area of DNA that previously could not be bound by that oncogenic transcription factor? So in order to answer that question, we used an electrophoretic mobility shift assay. So this is a very simple assay where you take either wild-type DNA, mutant DNA, and you basically mix it with the protein you think it interacts with. And if it does, it will alter the banding pattern on a polyacrylamide gel. So when we did that, we found that the wild-type 5' UTR did not bind at all to MYC and MAX. However, the mutant did. And what's particularly gratifying is that when we washed this out with an unlabeled competitor, that signal went away. So what this means is that a single point mutation with this 5' UTR created a new promoter region that was otherwise unrecognized by MYC before. So for the last example, I'm going to show you basically a translational hit of an oncogenic cell cycle regulator called CKS2. And this was also an upregulation. There's also a C to T mutation. And so what we see here is, a diagram of what the CDS looked like relative to the 5' UTR. And the mutation happened 65 bases upstream. But if you look closely, what happened was that it created a C to a T, and that trinucleotide context is a new start codon. And so what would be predicted is you would get an N-terminal extension of CKS2 if this actually is happening in the cell. So in order to determine if this is the case, we went ahead and used CRISPR-based editing to engineer this mutation, this precise mutation, into cancer cells. And what we found was that indeed it led to a significant increase in the protein levels of an N-terminally extended form of CKS2 without having any effect on the wild-type levels of CKS2. And very interestingly, there was no effect on RNA abundance, so this was an effect at the protein synthesis level. And what this demonstrates is a single point mutation at the RNA side of a 5' UTR can actually not only increase protein levels, but even change the identity of the protein. Now, given all the sequencing data we had, as well as the patient-related data, we sought to determine if there's any outcomes association with 5' UTR mutations. And so we basically took the data set and turned it into a localized and metastatic, and we did gene set enrichment analysis, and what we noticed was that the MAP kinase signaling pathway was enriched in patients with metastatic disease. And when we saw this data, we immediately thought of Hong Wu's paper as well as Koryabate Shen's paper showing that if you basically hyperactivate MYC, that leads to metastasis in mouse models. And so we looked back at our patients and we asked, well, what's the metastatic state of them? And we found that if they had 5' UTR mutations to the MAP kinase signaling pathway, they were 2 times as likely to present with metastasis at diagnosis. Now, this is very intriguing data. We think there's still actually a lot to do because we had to actually answer the cellular impact of these mutations. And I actually had a graduate student who joined the lab and was like, well, actually, I want to study the 3' UTR. And so she has actually identified tens of thousands of mutations of 3' UTR. And she's actually, for that, you actually study RNA stability, and she's created a new massively parallel reported technology to actually study this in an unbiased manner. I hope to report this to you in the future. So I want to thank the patients and their families for which this work would not have happened, and obviously the years of support from PCF. I'd like to thank my lab, and also I'd like to thank the Nelson Lab. The Ha Lab is not listed here, but they were very important for this. The Eisman Lab from the Fred Hutch, as well as the Chen and Carver's Lab from Memorial Sloan Kettering, and the Sharp Lab from the ICR. Thank you very much. Happy to take any questions.
Fantastic work. It took me back to graduate school looking at those mobility shift assays.
MSAs, yeah.
I had a question about the translation part of the talk. So I was wondering if you saw any effects on ribosomal RNA translation, because you need more ribosomes to do the translation. So is AR knockdowns affecting ribosomal RNA, or is it just messenger RNA?
Hey, Jelani, that's a really, really great question. We have, as we've looked at the field of translation, how much is known in prostate cancer, there is a lot known, but there's a lot not known. And I'd say the non-coding RNAs, like our RNA themselves, is a very wide open field. Because, you know, the field has rightly focused on Mallet and Schlapp, has focused on microRNAs, but I think our RNAs themselves, it's really not known. And how they change with androgen receptor activity is really not known. We've also developed an interest in tRNA biology too, because it's just not known. Exactly.
Yeah, and the other thing was, DNA is found in the mitochondria, right? So there's translation there. It's like 1% or something.
Yep.
But I wonder if this is affecting mitochondrial translation.
I'm almost sure it does, and I'm almost positive it's very different.
Yeah.
You know, I'm almost positive it does.
Hey, Andrew.
Yes, thank you.
Beautiful talk. I have a question regarding your first part about translation, and you conclude that AR negative or AR low is more sensitive to the translation inhibition, and your data show beautifully that the tumor growth is inhibited in the AR low model, but not the AR positive model. So can you comment what happened to the tumor that has like both AR positive and AR low cells? Would be like your compound might select out the AR positive cells, or they might kill both?
Yeah, I think in that context, if I had to look at our data, very carefully, I'd say that it'd probably kill one over the other. And so you might then actually get a selection for an AR positive population, which is more easily druggable nowadays than it would be to drug the other.
Yeah,
that's a great point. Very great point.
Hi, great talk.
Hey, Steve.
Your finding for AR regulation of 4-ABP1 is really surprising, because you'd expect in normal prostate that wouldn't necessarily happen. I'm wondering, this is probably all done in tumor cell lines. Have you looked at normal prostate, whether AR is positively regulating 4BP1 in normal prostate, or is this a switch that
Right.
occur in tumors?
That's a very good question. So the question basically is AR in normal prostate and whether or not this is signaling axis that's there. We haven't looked carefully, but I have speculated, and I speculated in just thinking about this problem, that it's possible that AR, when it's intact, right, and it's signaling in a normal prostate, this in the mouse, that it's mainly used for differentiation in that context. And because of that, it wants to hold down translational signatures that can regulate proliferation. So as a result of that, it could have a use in kind of dampening the proliferative potential of a cell in that context, at least from a translation perspective.
That would be interesting. I mean, it does regulate a lot of proteins also that are
Yes.
directly AR regulated.
Absolutely, absolutely.
Thank you.
Thank you.
Yeah, Andrea Limonti, I just want to ask you, beautiful talk.
Thank you.
By the way, so you show very quickly that you have an inhibitor for BP1, if I'm not mistaken, right? It was a pharmacological inhibition?
Right, yeah, so we used you probably know the Jerry Peltier's compound, the 4E1R cat.
Yeah.
So it's the eIF, yeah, it's basically a 4E4G inhibitor.
Yeah. So the question is, the effect of that inhibitor are also mediated to the tumor immune response, or did you check what's happening to the immunolandscape of this
Right.
tumor?
That's a great question. We did not, in this study, determine whether or not there was a non-cell autonomous effect from that drug. Now, those experiments were done in immune compromised mice, so it wasn't the perfect example, the perfect place to do it. However, we have a preprint right now where we've done single-cell RNA-seq in this progression model, and we see dramatic shifts in the immune landscape. There's no question about it. So I would not be surprised if there will be an interface there. And I think it'd be great to talk to Daniela actually about her data, because I think there could be some synergy there.
Go ahead, Jonathan.
No, I was going to say, Excelsior talk, is it fair to conclude that all prostate cancer may be a c-MYC disease of e-box creation?
Yeah, I mean,
UTRs? I mean, because if anyone, thank you for citing Hongwu's CapCure-funded work, and for those who might not know it, I mean, it's explosive. It's an explosive metastatic phenotype, right?
and Corey's model too.
No, in the same, but the question is, should medical oncologists start to think about prostate cancer as e-box, basically a disease of e-box creation?
I mean, I think the e-box creation part needs more investigation. We did not have numbers, I think, in ours to be able to adequately answer that. But like, for instance, I was talking to Colin Pritchard. He was telling me about 5' UTRs he's looked at through Oncoplex. And I think the data is definitely mounting. And what I'd like to do is just very carefully look at those and see how much e-box creation you have there. I think we limited our e-box look to only the 5' UTR, which is, proximal to the transcription start site. And the big question is, if you go even more upstream, how many e-boxes can you be creating there? And that would create a completely novel transcriptional landscape compared to like a normal plain DNA.
Following Jonathan's question, over here. Hey, Andrew.
Hey, what's up?
Lovely talk. So how recurrent were the alterations that you were observing creating these e-boxes? Because generally speaking, recurrent mutations in the non-coding regions of the prostate cancer genome are relatively infrequent. So are either for literal exact copies of the same alteration, or for ones that were predicted to have the same relative function, how recurrent are these?
Right, so I mean, the truth of the matter is for 5' UTRs and looking at our small data set, I'd say recurrency within the 5' UTR is relatively limited, okay? There could be 2 to 3, we've had up to 10 for one particular hit. So I think it's relatively limited. We really used this method to kind of understand the biology of 5' UTR and using cancer as kind of the sieve to help us understand what might be important. I would have to say though, just looking at the PCOG study, which obviously is a very important study, but with the way they talked about the non-coding space was like, oh, we didn't see any hotspots in the 5' UTR. And I'm not exactly sure that that's completely relevant, right? At the end of the day, it is within the tumor cell and which clone actually has something that helps it grow. If it likes SGF7, it's going to take off. So I think it's really, I think there's a big point to be understood there,
So, hi.
Yes?
I have maybe.
Sure.
Very inspiring talk. I have a quick question. So about the 5' UTR mutations, does this assay consider some large-scale changes like deletions, insertion beyond like point mutation? Another related, in our study, we also saw a lot of splicing changes in 3' UTR and 5' UTR. How does that take into this kind of functional things?
Right, so that is a very important question. We did not look carefully at splicing because in that case you'd have to look at the mRNA. In terms of indels, there were not that many indels. But again, we use Mutect and then Strelka. Strelka has indels, but it's like 10% of the total population of all the mutations. So there weren't that many. With that said, though, I also, as we, this project took like 5 years to do, and as we were working through it, I was like, man, why do we start with prostate cancer? And the reason why is because the amount of mutations in prostate cancer is relatively little compared to, let's say, melanoma or bladder cancer, But we were fortunate that we were able to find some hits and validate them. But I think those are very important questions, and it's completely unexplored and should be looked at closely.
Thank you.
So very stimulating talk, and I have a specific question. So in your AR-low environment where you see upregulation of a specific set of, the translation of a specific subset of transcripts, you identified an enriched element. Any clues as to the identity of what binds that element?
That's a great question. So you're asking the million-dollar question in terms of a cis regulatory element and trans acting factor and that interaction. We don't yet. What we've done now, though, is that we have actually created a reporter, a stable cell line that has a reporter for that guanine-rich translational element. And our plan basically is to subject that to the, and this is a reporter, so it'll actually read out, I think it's GFP. So the idea here is that we're going to subject that to the Brunello CRISPR library to try to find what that transacting factor is.
Can you use it as a bait and cross-link?
There's different debates on how to do this. Either you can do it functionally, which would be like through a CRISPR library. The other way would be to actually use it an MS2 stem loop, pull the thing down, and then mass spec for the proteins. We haven't tried that yet, but that's something we're interested in doing as well. And I think those would actually be very complementary. Yeah. Thanks for asking the question.
Yeah, sorry to come between everyone's dinner, but
I think I did that officially.
No. So this was a beautiful talk. So
Thanks, Corey.
You, of course, know our paper quite well. And in our study, we found that MYC is not sufficient for metastasis. As you know, it actually had to collaborate with RAS, and we showed that both in the mouse model, RAS activation, both in the mouse model and in human cancer. Very striking relationship. Although MYC was the top pathway that came up, it was always MYC and RAS collaboration. So how do we understand that in light of what you showed us today about MAP kinase signaling, et cetera?
Yeah, I was, after your paper and Hongwu's paper came, I was blown away by it. And then I remember seeing a bunch of other stories, thinking to myself, what are the things that influence MAP kinase signaling pathway? What upregulates it, right? Like, there's not a clear, genetic hit, there's no KRAS mutation or anything like that. So I was excited to see it pop up on the 5' UTR side, right, as a potential mechanism for upregulation of that. But I would say, yeah, I mean, to me, our data would suggest that in advanced cancers, right, we can see mutations that could allow transcripts to be under the influence of MYC, and at the same time, they can also acquire mutations within the 5' UTR that activate the MAP kinase signaling pathway. And maybe in that context, a combination of the two, and that leads to the aggressive disease that we see. Yeah.
Okay.
Thank you. Appreciate it.